Hello students. So we are talking about the catalysis, and you know that almost for every chemical reaction we need one or the other catalyst. One or the other catalyst is required, and no doubt when catalyst is present there, it increases the rate of reaction. It speeds up the rate of reaction. Now similarly, lot many chemical changes are taking place in our body also. Lot many reactions are going on in each and every cell. So for those also we need some catalyst. So now today we have to discuss about the catalyst which are needed in living systems to carry out the reactions, to carry out the chemical reactions. Means here we have to talk about biological catalysts which are called as enzymes. So today we have to discuss about enzymes. And interesting, these enzymes are so important. I just want to share one thing with you. When we eat the food normally get digested within a span of 4 to 5 hours, generally maximum 4 hours. But remember, if there is no catalyst to digest that food, that meal, can you imagine how much time it will take? So science tells it will take approximately 50 years to digest a single meal if no enzymes are present there. So these enzymes are so vital and so important in our body. So those enzymes we have to discuss with you right now. And what are the, how can you define them? These are the biochemical catalysts. Bio means produced, chemicals means these are the chemicals which are produced in living systems. That's why they are called as the biochemical catalyst. Remember, chemically these are globular proteins. These enzymes are proteinaceous in nature. You know, the important organic compounds needed by body are classified as carbohydrates, fats and proteins. So out of those, these enzymes are globular proteins, globule shape. These are spherical bodies, these are which immediately dissolve in water to form a colloidal solution. So these are the, and they have high molecular masses. So these are enzymes we have to talk. And remember, just see now, in a typical cell, it contains almost 3000 catalysts. Just imagine, in each and every cell of the body, there are approximately 3000 catalysts to carry out all the normal functioning of a single cell. So see how important these enzymes are in our body, in the living systems. First of all, what we will do, we will talk about some important characteristics of these enzymes. The very first point is there, I just told you, I am repeating, they form colloidal solutions in water. Because these are proteins, because these are globular proteins, you add them in water, you will get a colloidal solution. Next, highly specific in nature. Now compare with catalysts. Catalysts are also highly specific in nature. Enzymes are they are also highly specific in nature. Means a particular enzyme can catalyze a particular reaction only. Can catalyze a particular biological reaction only. For example, we want to digest lipids. We need an enzyme called lipase. Suppose we want to digest starch. We need another enzyme. This is called amylase. And remember, I am just sharing one thing. In almost at the end of every enzyme, you will get A's. Lipase, amylase, protease, diastase, maltase, sucrase. So this A's is for enzyme, and this first word is for the, you can say, substrate or compound on which that enzyme is going to attack on. So these enzymes are highly specific. For every different substance we need a different enzyme. Now similar to catalysts, they have high efficiency. They speed up the rate of reaction. They catalyze the substances in the body to a great extent. They can increase the rate of that reaction very high. You see, to an extent of up to 10 raised to the power 20 times. So high. means reactions become so fast. You can compare. I just told you, okay, without enzyme, meal can take 50 years to digest, and in presence of enzyme, you take almost 3 meals a day. Means after every 4 hours it get digested. Similarly, now what else? It needs an optimum temperature. An optimum temperature is best the human body temperature, means enzyme works best at a temperature of 37 degrees Celsius. Best. This is the optimum temperature for working of enzyme. But as and when temperature reaches to 70 degree or beyond it, enzyme activity get destroyed. Enzyme does not work anymore. So optimum temperature 37 degrees Celsius. Likewise, enzymes need an optimum pH also. pH means medium should be acidic, basic or neutral whatsoever. So enzymes work best at an or needs an optimum pH. An optimum pH for an enzyme to work best is 5 to 7. This is the pH value between which enzyme can work, and the best again it is 7.4, where enzymes works best to it. So these are certain important characteristics. As in case of catalyst, we studied there are catalytic promoters. Similarly, for enzymes, for increasing the activity of enzymes, we have coenzymes and we have activators also. In brief, I want to tell, normally when you say coenzyme, these are organic molecules. You know, enzymes are proteins. So coenzymes are non-proteins which get connected with enzyme and enhances the enzyme activity. Like some enzymes, with some enzymes, vitamins get connected and they increases the enzyme activity. These are coenzymes, organic but non-protein. And then what are activators? They also increases the enzyme activity, but these are generally metal ions. Like you see, these metal ions like Na positive, Cu dye positive, Mn dye positive, cobalt dye positive, etc. So many are there. But these are, when you say activators, these are metal ions. When you say coenzymes, these are organic molecules which are non-protein in nature. Like for example, if NaCl is added, so because of Na positive ion, enzyme amylase which has to work on starch, it works better. It works good. Right? Now similarly, we have enzyme inhibitors. So when we talk about enzyme activator which increases the enzyme activity, we have enzyme inhibitors which decreases the enzyme activity. I will discuss it more here, but these are some of the important characteristics of enzymes. Now, next topic, how these enzymes actually work. Mechanism of enzyme action we have to talk. As you know, in homogeneous catalysis, intermediate compound formation theory discussed. In heterogeneous catalysis, adsorption theory discussed. Similarly, enzyme they work, they also have some mechanism. So here we have to talk about lock and key model of enzyme action. I just told specificity. Enzymes are highly specific in nature. Now this enzyme action, why do we call it as lock and key mechanism? As for a specific lock, to unlock it, we need a specific key. Similarly, a specific enzyme works on a specific substrate. Why is it so? Now I am just sharing. Actually, enzyme for action, they have certain active sites. They have definite shape. Now when enzyme is added to any reaction, only that substrate can, you can say a substrate will fit itself in this active site. So remember, let us consider we have 3 different substrates. So only that substrate can fit itself whose shape matches with the shape of enzyme, matches with the shape of active sites. And you see, active sites contain certain functional group. Like NH2 they contain, like COOH they contain, like OH etc. Such groups are present there, and as and when substrate come to combine here, they form bonds with this substrate. So 2 things are there. First, a specific substrate can come to combine whose shape match with the enzyme. And secondly, when substrate comes to combine, it forms bonds because of presence of these groups. And what bonds? Hydrogen bonds, van der Waals forces, and this results in the formation of a enzyme substrate complex. Enzyme substrate complex. Now this enzyme, and remember, a specific substrate can combine. Now when this enzyme substrate complex is formed, now later on this enzyme substrate complex break to form product, and enzyme is regenerated for reuse. So this is lock and key mechanism of enzyme action. And you see, I just told you enzyme inhibitor. How does it deactivate this enzyme? What it do? It comes to combine with this enzyme inhibitor, comes to combine with this enzyme and deshape it so that substrate cannot combine with it. This is how enzyme inhibitor work here. So now the important thing, enzymes, what are the important characteristics they show and what are the mechanism of this enzyme action. It's really very important. Listen it carefully. You can watch it again. Thank you.